Maven International Limited is a commercial research and innovation organization which develops, manufactures, and distributes its own novel sustainable agri-tech products. We use biorational green chemistry strategies to minimize the use and generation of substances harmful to the environment with technological approaches for preventing pollution while remaining fully effective. I would like to introduce ELICITEX, our new sustainable indirect pest control product in our P range. In the diversity of life on Earth, nature has evolved an extraordinary plant defense system which, even though it is unseen, exists all around us. Our planet's most abundant building blocks are natural biopolymers found throughout Earth domains. From the deepest oceans, the densest forest to your own gardens. Meet these biopolymer elicitor molecules designed by evolutionary process and brimming with the potential to revolutionize the entire agricultural industry. These truly remarkable compounds can be used for safeguarding our most precious crop resources. The plant immune system is composed of surveillance systems that recognize multiple different phytopathogen elicitor molecules. These signal plant cells to switch from the production of plant primary metabolites such as carbohydrates, lipids, and proteins, which are directly involved in growth and development, to the production of plant secondary metabolites, which are involved in plant defense and result in protection from potential harm. In plant biology, elicitors are extrinsic or foreign molecules often conserved essential structures in pathogens and pests. These conserved pest biopolymers, referred to as pathogen-associated molecular patterns, are recognized by complementary plant receptor proteins called pattern recognition receptors. Upon first contact with specific cell receptors, the plant is effectively tricked into believing it is under attack. These molecules cause a cascade of desirable reactions, inducing plant protection from pest predation while increasing its nutrient uptake and accelerating growth. By understanding this naturally evolved plant defense system, Maven has developed a whole range of remarkably effective crop protection elicitation molecules which are biodegradable, have biocompatibility and atom economy with multifunctional efficacy. We believe these strategies will open up domains of opportunities for the future of sustainable, innovative, climate-smart agriculture and have the capacity to make significant global impacts. So let us look at how these remarkable elicitation molecules work. ELICITEX contains a proprietary cocktail of food-grade oligopolysaccharides and oligopolypeptide compounds, including modified keto-oligosaccharides, beta-glucans, pectin, inulin, and cyclodextrins, which all act as highly efficacious elicitors. Maven has modified the structure of these compounds for optimal molecular weight, biopolymer chain length, and level of deacetylation to achieve maximum plant receptor affinity. These plant receptors are able to recognize the molecular patterns of elicitors, like a lock recognizes a key, and when the plant receptor is occupied by its specific elicitor, it triggers the production of second messengers and a signal transduction pathway inside the plant cell. This results in an intracellular defense cascade with enhanced transcription factors and upregulation of gene expression with increased rate of DNA transcription into RNA, which is then translated into proteins which function as enzymes and catalyze the production of bioactive plant secondary metabolites in biosynthetic pathways. These truly remarkable compounds act as defensive molecules which protect plants from pest predation and disease, rather like a vaccine for plants. We use multiple elicitors to increase gene expression of multiple enzymes in multiple plant secondary metabolite biosynthetic pathways. Most chemical reactions do not occur spontaneously in plant cells at their physiological pH and temperature and require enzymes to allow reactions to occur which would otherwise be impossible in a normal biological timescale. So how do they work? They speed up chemical reactions by binding to specific reactants or substrates. Binding occurs at a region called the active site. Once the substrates bind to the active site, they form an enzyme-substrate complex. As the enzyme and substrates interact, the conformation induced by the enzyme allows for a different reaction pathway with a lower and more energetically favorable reaction barrier. Chemical bonds are broken and formed, leading to the synthesis of a new molecule. This is referred to as the product. Once the reaction is complete, the product is released from the active site and the enzyme returns to its original state and is free to react again with another set of its specific substrates. Different plants have evolved to protect themselves in different ways by producing different plant secondary metabolites, including beneficial compounds such as anthocyanins in strawberries, giving its berries properties such as their red colour. And in chili peppers, capsaicinoids are synthesized and are responsible for their pungency, and carotenoids are produced which contribute to their colour and nutritional density. All plant secondary metabolite biosynthetic pathways are sequences of enzyme-catalyzed chemical reactions within plant cells which convert a substrate into a desired product. They involve multiple steps, each catalyzed by a specific enzyme, transforming the substrate into various intermediates before eventually forming the final bioactive plant secondary metabolite end products. Each plant can be considered to be a cellular, compartmentalized metabolic factory for the production of these truly remarkable chemicals. We use strategies of multiple elicitation to upregulate the enzymes in different biosynthetic plant secondary metabolite pathways. We also supplement common metabolic precursor substrate intermediates as a precursor feeding strategy for increased metabolic flux and production of these high-value plant secondary metabolite phytochemicals. This elicitation process is also primed with proprietary biostimulation molecules to enhance nutrition efficiency, abiotic and biotic stress tolerance, and crop quality traits, regardless of the plant's nutrient content. ELICITEX thus represents three synergistic, sustainable, climate-smart strategies in one biorational product, and by combining elicitation, precursor feeding, and biostimulation to enhance the synthesis of endogenous plant secondary metabolites, we can use ELICITEX to unleash the power of nature's potential. So what are the benefits of using ELICITEX? It provides fast and long-acting indirect control of plant pathogens and industry pests by induction of endogenous defense plant secondary metabolites. For example, ELICITEX induces disease resistance in Solanum tuberosum or potato plants by boosting the plant's own innate and adaptive immunity. It is active against potato soft rot Pectobacteriaceae and effectively induces endogenous potato defense activity against the fungal pathogen Alternaria solani that causes early potato blight and the omicet Phytophthora infestans that causes late potato blight. ELICITEX also prolongs the endodormancy of the lateral tuber bud meristems or eyes and delays sprouting to achieve safe storage of potatoes with bud control and therefore reduces storage loss. It is not harmful to beneficial organisms such as pollinators like bees or beneficial organisms which predate on plant pests in biological control programs. So, it fits well with biological control programs and as part of integrated pest management systems. ELICITEX acts as an antitranspirant and significantly reduces water use by abscisic acid induction, which has downstream effects on stomatal conductance. ELICITEX can also be used to precondition mother plants to improve cloning success. It is an effective growth promoter and improves tolerances to osmotic, salinity, drought, and extreme temperature stresses. Its active substances are food grade and can be used up to harvest without human toxicity, have no residual activity, and are safe for use on all edible crops and ornamental plants, and it also rapidly biodegrades. ELICITEX therefore provides biorational security to users and the environment. However, the marvels of these plant secondary metabolites extend far beyond this. When ELICITEX signals the rapid and highly efficacious induction of plant secondary metabolites, which include anthocyanins, terpenes, phenolics, capsaicinoids, and carotenoids, it increases crop nutritional density and improves organoleptic properties including flavour, odour, colour, and therefore customer satisfaction. The induction of plant secondary metabolites also has the added benefit of extending the shelf lives of harvested crops by delaying crop decay and thus reducing food waste and in particular prolonging soft fruit firmness. Here you can see the effects of ELICITEX on the left and no treatment control on the right. The normal shelf life of strawberries is 4 days. The defensive plant secondary metabolites induced by ELICITEX prolongs post-harvest shelf life by preventing powdery mildew, grey mould, Botrytis cinerea without adversely affecting taste or smell. This product can therefore reduce food waste by prolonging post-harvest shelf life for up to 2 weeks. Some fruits are non-climacteric, meaning that they do not continue to ripen after being harvested and therefore need to be harvested at their optimal ripeness, as they only ripen while being attached to the plant or vine. This can be commercially important if fruit ripening time is prolonged. Examples include berries such as strawberries, raspberries and blueberries, as well as citrus fruits such as grapefruits and lemons, and also cherries, chili peppers and coffee plants. ELICITEX can upregulate endogenous plant secondary metabolites and induce the rapid ripening of these non-climacteric fruits. Here you can see the effects of ELICITEX on the left and no treatment control on the right. ELICITEX treatment induces the rapid ripening in strawberries and production of the plant secondary metabolite pelargonidin 3-glucoside, a red anthocyanin pigment, within 16 days post-anthesis, which is the period when the flower is fully open. Here you can see similar effects of ELICITEX on the left and no treatment control on the right in chili peppers, another non-climacteric fruit. ELICITEX treatment induces the rapid ripening and production of the plant secondary metabolite carotenoid, a red-yellow pigment within 40 days post-anthesis. This allows for earlier peak ripeness and earlier harvest. The marvels of elicitation do not end there. Remarkable human health benefits have been demonstrated following the ingestion of plant secondary metabolites in terms of the prevention of chronic disease. Plant secondary metabolites are known to have significant nutraceutical and human health benefits, including anti-obesity, anti-diabetic, anti-cancer, neuroprotective and cardioprotective effects. So why not try ELICITEX? The novel revolutionary product from Maven, which unleashes the power of nature by harnessing the advanced biorational technology of evolutionary biology. It is available as an ultra-concentrated liquid suspo emulsion for large-scale dilution in commercial tank mixing, or as a ready-to-use pre-diluted ELICITEX MIST. For further information, please scan the QR code. At Maven, we are fully committed to the continuous evolution of elicitation technology, so join us as we will unlock the boundless potential of these remarkable biomolecules, harnessing the power of nature for her own preservation. Maven commits to a triple bottom line policy which focuses as much on social and environmental sustainability as on economic profitability. Our mission statement is to continually deliver translational cutting-edge innovation through proof-of-concept research from the laboratory bench to the plant bed, linking green chemistry science to agronomy and placing people and planet before profit. Maven thanks you for listening.